Namaskara, Namaskara, Namaskara. So in this video we will discuss what is oxygen hemoglobin dissociation curve. Fine. Either you call it oxygen hemoglobin dissociation curve or you call it oxygen dissociation curve. You can see this topic, it's student feel it's some kind of complicated topic and they really not understand it 100%, and they used to see it in the topic breathing and exchange of gases. So this oxygen hemoglobin dissociation curve or oxygen dissociation curve, it's kind of a graphical representation of relationship between binding of oxygen with hemoglobin. Or same thing I can say in another way, that it is graphical representation of relationship between partial pressure of oxygen and percentage saturation of hemoglobin. Try to understand it what exactly it's saying. We all know that hemoglobin present in the red blood cell, it's a red color pigment, isn't it or not? The hemoglobin have two part. First part heme, second part globin. What is color of hemoglobin? Red color, right? This hemoglobin heme part is a complex ring, that is porphyrin ring, and this porphyrin ring have Fe2 plus ion. This Fe2 plus ion actually interact with oxygen molecule. Okay, in one hemoglobin molecule total 4 heme are present. Then they have next part, so that means if they have four heme, that means they can bind with total 4 oxygen molecule. And same thing you can say in another way, they can bind with 4 oxygen atom, 8 oxygen atom, yes. And then they have globin part. In this globin part total 4 polypeptide chains are present, 2 alpha polypeptide chain and 2 beta polypeptide chain. Alpha polypeptide chain have 141 amino acid and beta polypeptide chain have 146 amino acids. Fine. So this is what hemoglobin, adult hemoglobin. Fine. Now when this adult hemoglobin bind with oxygen, it have 4 place for binding. I'm making like a place, like binding site imaginary diagram here. First oxygen will bind here, then oxygen will bind here, then oxygen will bind here, then oxygen bind here. Total 4 oxygen binded with the hemoglobin molecule. So when the all oxygen binded with the hemoglobin molecule, I can say that it's this hemoglobin molecule saturated with oxygen. Now you imagine in your body total 100, suppose total 100 molecules of hemoglobin are present. If 50 molecule of the hemoglobin binded with oxygen, can I say like that okay, your 50% of the hemoglobin saturated with oxygen? Yes. If 75 molecule binded with hemoglobin, I can say that okay, your 70% of the hemoglobin, 75% saturated binded, so I can say your 75% of the hemoglobin molecule saturated with oxygen, right? For that a particular partial pressure is required or not? That is what we will see here. What is relationship of partial pressure of oxygen and percentage saturation of hemoglobin? Percentage saturation of hemoglobin. This percentage saturation of hemoglobin, you say, or you call it oxyhemoglobin. Okay, fine. So we will see it here, right? Okay, students, when scientists study it, one axis they take in partial pressure of oxygen and another axis they take in percentage saturation of hemoglobin, they observe one S shape, they observe one sigmoid curve. Now in experimental study, they observed that approximately at 25 to 30 mmHg, yes, a value between 25 to 30 mmHg, you can take it 26, partial pressure of oxygen at 25 to 30 mmHg partial pressure of oxygen, half of the hemoglobin molecules saturated with oxygen. I will call this value the partial pressure of oxygen at which half of the hemoglobin molecule saturated with oxygen. I'll use the word P50 for that. Okay. And then we observe that a value between 95 to 100 mmHg, all molecules of the hemoglobin saturated with oxygen. Clear, students? Okay. This is what something we got in experiments and all. So we got one sigmoid curve. We got S-shaped curve, sigmoid curve we got. Fine. Now it is observed that the binding of oxygen and hemoglobin is not merely regulated by the partial pressure of oxygen, but it have great role of other factor also. Like you see the Indian marriages, you see Indian marriages, suppose this is a boy, okay, and this is a girl. Their marriage, their alliance, their association, it will be not only decided by boy and girl, it will be decided by society also, it will be decided by family also, it will be decided by father and mother also, isn't it or not? So we have other factor also for binding of hemoglobin with oxygen. And what is that, students? That is hydrogen ion concentration, that is partial pressure of carbon dioxide, that is pH, that is temperature. Fine. Students, scientists observe that whenever in a medium we increase hydrogen ion concentration, the curve will shift right side. Right side means right hand side here. Okay, you see here. Shifted right side or not? Here. Shifted right side. So it is observed that when hydrogen ion concentration increases, hydrogen ion have direct relationship with the pH also. pH is negative logarithm of hydrogen ion concentration. So either when hydrogen ion concentration increases or pH decreases, this curve shift right side. Actually high hydrogen ion concentration or low pH, it will cause dissociation of more and more oxyhemoglobin into hemoglobin and oxygen. They will not promote binding of hemoglobin with oxygen. In fact, they will promote by dissociation of hemoglobin with oxygen, right? So this is the factor, high hydrogen ion concentration and low pH is the factor that will not support binding here. In fact, they will support dissociation. They will create disturbance in binding. Okay. Then we observe that when partial pressure of carbon dioxide increases, the curve shift right side. This high partial pressure of, obviously the when partial pressure of carbon dioxide increases, they will somewhere, because carbon dioxide also they bind with the hemoglobin and they form carb amino hemoglobin. So if their concentration increases, they will cause dissociation, and it is observed also in the tissues where partial pressure of carbon dioxide is high, that is 45 mmHg, they promote dissociation of oxyhemoglobin into hemoglobin and oxygen. And then it is observed that when temperature increases, the curve shift right side. It also affect, it's also disturb binding of hemoglobin with oxygen. And then see this process, this all process of binding of hemoglobin and oxygen is happening within the red blood cell. In the red blood cell, lot of glycolysis is going on. Glycolysis, is the 10-step chemical reaction in which glucose, one molecule of glucose break into 2 pyruvic acid. During this process of glycolysis, one intermediate form, that is 2,3 bisphosphoglyceric acid, 3 carbon compound, it is 2,3 bisphosphoglyceric acid. It is observed that whenever 2,3 bisphosphoglyceric acid concentration increases, the curve shift right side. That means this this 2,3 bisphosphoglyceric acid bind with hemoglobin and they will not allow oxygen to bind. Yeah. So obviously it creates disturbance. That's why the curve shift right side. Fine. Then we change the condition. Scientists, then they change the condition. What they did, yes, they changed the condition. They decreased the hydrogen ion concentration. They decrease hydrogen ion concentration. They observed that when hydrogen ion concentration decrease, the curve is shifting left side, means left hand side. Now you see, when the curve is shifting left hand side, we need less partial pressure for 50% saturation. Like if you put one line here, vertical, it will come somewhere here, isn't it or not? So partial pressure P50 decreased or not, right? And you see, for 100% saturation, as compared to here, we need less oxygen. That means whenever hydrogen ion concentration decreases, it support binding of hemoglobin with oxygen, and the curve shift left side. Whenever partial pressure of carbon dioxide decreases, whenever pH increases, whenever 2,3 diphosphoglyceric acid, 2,3 bisphosphoglyceric acid decreases, whenever temperature decreases, the curve shift left side. They promote binding of hemoglobin with oxygen, right? This is what scientists observe during study of oxygen hemoglobin dissociation curve. Now student may ask one question. What question they ask students? Sir, why this curve is S shape? Why this curve is sigmoid shape? So it's showing one clear pattern, students, it's showing one clear pattern that starting the binding of oxygen with hemoglobin is slow. See here, starting binding of oxygen with hemoglobin is slow, and then it's boomed suddenly. The rate of binding of oxygen with hemoglobin boomed suddenly, and then at last they came at a particular saturation point. They came. Actually, it have a direct role. This S-shaped curve have direct role of nature of oxygen, nature of binding of oxygen with hemoglobin molecule. See here, students. One hemoglobin molecule have 4 heme. That means 4 oxygen have to bind. So when oxygen will bind, this kind of binding is called cooperative binding. Cooperative binding. What is this cooperative binding? You see, we'll call it cooperative binding. Like we have a molecule of hemoglobin and it have 4 heme, one by one oxygen have to bind. First one oxygen binded. When this first oxygen bind, it promote binding of next oxygen. So next oxygen have more affinity to the oxygen hemoglobin molecule. Now this 2 oxygen molecule binded, this molecule is 2 oxyhemoglobin now. So they, for third oxygen molecule, 100 of the times affinity to the hemoglobin increases, right? And when last oxygen molecule bind, you see here, it have highest affinity to the hemoglobin. So that's why, so this binding, so this last oxygen molecule binded, these all 3 are facilitating binding here, right? And as see, starting it is slow because we have one oxygen which is binded with first oxygen binded. Now this first oxygen, is promoting binding. You see here, So that's why the affinity increased. Affinity increased when fourth oxygen binded, it's saturated. That's why it's so S-shaped curve, sigmoid curve. Now I have a question, students. Fetal hemoglobin, the hemoglobin who is present in the fetus. The fetus observed that the fetal hemoglobin have more affinity to oxygen. They can carry more than 30% of oxygen than the adult hemoglobin. Like she's in the mother, the baby is present in the mother's womb, as compared to mother, he or she have 30% of more oxygen. More, they can carry 30% more oxygen. Okay, students, I have written also here, even they can carry 30% more oxygen than the adult hemoglobin. What is reason? Actually, fetal hemoglobin can split into two parts. For him, for him, obviously one hemoglobin molecule have for him actually. So for him we have, and then alpha 2 gamma 2 polypeptide chain. Compare with adult hemoglobin. Adult hemoglobin have 4 heme and 2 alpha and 2 beta polypeptide chains, but here you see 2 alpha and 2 gamma polypeptide chain. Because of this gamma polypeptide chain, they have less affinity to 2,3 bisphosphoglyceric acid. And because of less affinity to bisphosphoglyceric acid, they will not, this 2,3 bisphosphoglyceric acid unable to create any kind of disturbance within between binding of hemoglobin with oxygen, and that's why they have more affinity to oxygen. Fine. Now I have a question to you. If I will try to settle this hemoglobin F, fetal hemoglobin, so as compared to adult hemoglobin, please tell me the curve will shift it left side or right side? Obviously left side. Why? Because even the less partial pressure of oxygen also, they can saturate nicely, right? That's why this curve will shift left side. Fine. So this is about fetal hemoglobin, HbF. So this is what oxygen hemoglobin dissociation curve or oxygen dissociation curve. Remaining we will see in next video. Bye.